A very good morning to Doctor and my fellow friends. My name is Kasturi Dayalan and my group members are Eng Tianzer and Kashmira Sargunet. The title of the journal that we're going to present today is Propionibacterium freudenreichii thrives in microaerobic condition by complete oxidation of lactate to carbon dioxide. So for the problem statement of our study is to show the increased biomass formation of 4 species of food-grade propionic acid bacteria when they're exposed to oxygen, they're implicating functional respiratory system. So these are the content that we're going to talk in our presentation today. So let me go through with what is our aim. The aim of our study is to find the biomass formation and the metabolite production of 4 species of food-grade PAB strands when it exposed to various levels of oxygens. So as you can see, these are the 4 species of PAB we use in this study. The 16 strands of propionic acid bacteria used for the screening are classified from these 4 species. But the commonly used dairy isolated Propionibacterium freudenreichii was selected for an in-depth study of its response to oxygen. Okay, let me start with our introduction. Let me introduce what is propionic acid bacteria. Propionic acid bacteria are combined into the family of Propionibacteriaceae, genus Propionibacterium. Propionibacteria genus are divided into 2 main groups based on their habitat, which is classical and skin. And the skin is also known as cutaneous. Propionic acid bacteria is actually a gram-positive bacteria, which means they are non-motile and do not produce bacterial spores. And they are also catalase positive. They are also recognized as anaerobic or relatively anaerobic bacteria. As we know, PAB are very small and they take the form of spherical shape, which is cocci, under anaerobic condition, while in the presence of oxygen, they demonstrate in club shape. So the optimal pH of propionic acid bacteria oscillated around pH 7. As we all know, propionic bacteria species are mesophiles. However, they are resistant to much higher temperature because they can survive up to 20 seconds at 70 degrees Celsius. And their distinct feature are formation of propionic acid as a consequence of propionic acid fermentation, dependent on the coenzyme B12 and the high content of GC. GC stands for guanine cytosine, and they have almost 65 to 75% of this GC content. And last but not least, propionic acid bacteria can be isolated from soil, herbaceous plants, and the digestive tracts of ruminants. So that's all from me. Now I will pass the floor to my friend. Thank you.
Hi everyone, my name is Eng Tianzer. So let me continue with the methodologies. In this study, the main methodologies which is proteome analysis. After the 16 strands of propionic acid bacteria are undergo screening, the proteome analysis method will be used to analyze the particular strength. Proteome analysis is revealed that the abundance of protein involved in the aerobic and anaerobic electron transport change and major metabolic pathway did not significantly differ between anaerobic and microaerobic conditions. Proteomics is used to detect protein expression pattern at a given time in response to a specific stimulus, but also to determine the functional proteins network that exists at the level of the cell, tissues, or whole organism. Furthermore, let me to do the further explanation of the propione analysis. In this experiment, they use 16 Propionibacteriaceae species strains which can classify into 4 species of food-grade propionic acid bacteria, which is 3 acidic Propionibacterium acid Propionibacteriaceae, 5 acidic Propionibacterium jensenii, 2 acidic Propionibacterium threonii, and 6 Propionibacterium freudenreichii, which isolate from different dairy sources. All these strains were initially cultivated using the yeast extract lactate medium, which is YEL, containing L-lactic acid tryptans, yeast extract, and potassium dihydrogen phosphate. The pH was adjusted to 7 by using 5 mol of sodium hydroxide to autoclaving at 121 degrees Celsius for 50 minutes. The strains were stored in 30% of glycerol curvi at 80 degrees Celsius. In this study, we screened the response to various levels of oxygen of 16 propionic acid bacteria strain in 3 different conditions, which is aerobic shaking, aerobic static, and anaerobic. The first process of the protein analysis, which is culture condition initial screening. The strain were shred from negative 80 degrees Celsius stocked on yeast extract lactate medium plate. It incubate for 7 days at strict anaerobic condition using anaerobic gas and an oxygen catalyzed at 30 degrees Celsius in anaerobic jar. For aerobic incubation, single colonies were inoculated in 10 ml yeast extract lactate medium in 100 ml shake flask and incubate at 30 degrees Celsius static and shaking for 5 days. For anaerobic incubation, single colonies were inoculated in 10 ml yeast extract lactate medium in 50 ml junior tube. Anaerobic samples were incubated at 30 degrees Celsius for 5 days in anaerobic jar at strict anaerobic condition using anaerobic gas, and oxygen catalyzed at 30 degrees Celsius in anaerobic jar. The optical density was measured at 600 nanometers. Besides that, long-term batch cultivations are used to explore how evolutionary process of propionic acid bacterial strain work in heterogeneous environment where cell experience stirring speed at 300 revolutions per minute, the temperature that constant at 30 degrees Celsius, and the pH was controlled at pH 7. After the long-term batch cultivations, it will undergo the process of biomass quantifications. Biomass was quantified by measuring the cell dry weight concentrations. The weight sample will pass through the pre-weight membrane filter with a pore size of 0.2 micrometer by a vacuum filtration unit. The filter will dry and weight to determine the cell dry weight concentration in culture. Moreover, it will undergo the analysis of extracellular metabolite. Lactate, acetate, and propionate were qualified by high-performance liquid chromatography. Lastly, the proteome analysis, which is the proteomic samples preparation and analysis, is to determine the protein similarity among samples and review protein expressions pattern across the samples, and is clustered the samples by using the hierarchical clustering and k-means partitioning.
Hi doctor, I'm Kasturi here. Let me continue with the result. So we can see there are 2 graphs here. Graph A represents biomass production, while graph B represents acetate production for 4 species of propionic acid bacteria of cells growing in 3 different conditions. So from each data point represent the average of biological duplicates for each individual strands. So from this graph, there is a significant difference between the optical density value found between anaerobic and aerobic conditions. So from this graph, we can conclude that the availability of oxygen can enhance the biomass formation in static condition for all strands, although the strand-to-strand variation are enlarged. But for the high oxygen level in aerobic shaken condition, the biomass production become more lesser when compared to anaerobic condition. So there are several strands that were able to produce similar or higher biomass compared to anaerobic condition, and thus are more tolerant to higher oxygen level. That's all from me. Thank you.
Overall idea of this experiment is these bacteria, are they thrive well in microaerobic condition? So if they thrive well, what will happen? What is the product of this process they undergo? So for this one, let me explain to you. So here we can see two different conditions, which is anaerobic and microaerobic. And then here you can see concentration of organic acid, time in days, and then compound, that which we are focusing on. Green for lactate, red for acetate, while blue for propionic. So let me see here. As you can see from here, in growth phase 1, there is concentration of lactate slowly depleted. So as you can see from here, let me tell you, in both conditions, in whether it's microaerobic or anaerobic, the lactate concentration depleted. So it can be sure that at this phase, lactate consumption phase has been taken place. So focusing on microaerobic condition, the maximum there is a higher maximum growth rate and biomass achieved in lactate consumption phase, which is, let me explain to you more when we going on when I show you the proteomics data that we get from. So moving on to the anaerobic fermentation of lactate, it shows that there is average yield of cell driving compared to microaerobic conditions. So we can summarize that the production of propionate in microaerobic condition implies that the flux through the electron transport chain is limited or even stoichiometrically constrained by the availability of oxygen, causing the propionic production as the main electron sink. So in the same time, let me tell you, lactate acts as, in this phase, lactate acts as the carbon source. So what happened to the carbon source when it's depleted? So as you can see from here, let me move on to the next one. When the lactate depleted, there is production of acetate and propionate, which slowly increases, as you can see from these two charts. So at this growth phase 2, when the lactate is depleted, there is an initiation of propionate consumption phase, as you can see from here. The concentration of propionate is slowly depleted, so indicating there is a consumption of propionate is going on there. So a switch from propionate production and to consumption can be observed here from anaerobic to aerobic condition by this bacteria. So the E. coli switch the production of dissimilation of secreted acetate, which is also known as acetic switch. When its preferred substrate, it becomes limiting and electron transport chain is possible. So the name of this acetic switch, which is in growth phase 3, acetic switch can be derived from production to consumption of propionate when lactate is depleted in presence of electron acceptor in P. freudenreichii, the propionate switch. So let's move on to the third phase. So as I tell you earlier, at this growth phase 3, there is acetate consumption phase is going on. So there is no other, in this phase there is no other organic acid is detected. So from here it shows that there is a complete oxidation of acetate to carbon dioxide is happened. And then another thing that I would like to say, that the acetic switch occurs as the cells deplete in their environment of acetate producing carbon sources and begin to rely on their ability. So as you can see from here, I want you guys to focus on the maximum growth rate of lactate consumption phase and anaerobic lactate consumption phase in anaerobic condition. So as I told you earlier, there is a average yield of lactate consumption phase in anaerobic, which it is proven here, and microaerobic condition, there is a higher yield when it compared to other phases, including anaerobic condition. That's all from me. Thank you.
Hi friends, let me continue with the discussion. As I mentioned earlier, in this study we used 16 propionic acid bacteria strands to screen at various levels of oxygens. So the three conditions that I mentioned earlier is aerobic shaking, aerobic static, and anaerobic condition. So my friend will continue with further discussion. Thank you.
Let me to continue the discussion about this figure that show The metabolism of the lactate in the Propionibacterium freudenreichii in microaerobic conditions. Undissociated acids can be transported through the passive diffusions that the figure show the closed arrow across the membranes. Dissociated acid have to be transported by transporter that the figure show the dash arrow with the closed circles. Lactate is metabolized in three phases, which is a lactate consumption phases, propionate consumption phases, and acetate consumption phases. In the lactate consumption phases, lactate is metabolized to propionate and acetate. In propionate consumption phases, the propionate switch occurs and the produced propionate is metabolized to acetate. In acetate consumption phases, the acetate switch occurs and acetate is metabolized to carbon dioxide. Several options has been proposed for the production of the acetyl-CoA from pyrite, either a pyrite dehydrogenase complex that producing the NADH, or pyrite ferredoxine oxidoreductase that producing the reduced ferredoxines that catalyze the conversion of the pyrite to acetyl-CoA. Recently, a ferredoxine is act as the base energy conversion system has been suggested in propionate acid bacteria consisting of fixed ABCX, which oxidize reduced ferredoxines and chrono by reducing to NAD+. The entire complex fixed ABCX could also be reconstituted in our proteomic data, supporting the possible role of the pyrite. Ferredoxine oxidoreductase is the synthesis of the acetyl-CoA from pyrite and maintaining the redox balance for anaerobic growth on lactate. A pyrite oxidase that we can see the black dotted line that producing the acetyl phosphate from pyrite and phosphate while consuming the oxygens has also been identified in the genome of Propionibacterium freudenreichii, which may function as an energy yielding oxygen tolerance mechanism and which may explain lower propionate acetate ratios found in microaerobic conditions compared to anaerobic conditions in the Propionibacterium freudenreichii.
Hi everyone, it's me again. I'm gonna summarize the overall process that my friend explained to you earlier. So, uh, here we have fermentation of lactate, oxidative branch, and reductive branch, which is Woodward-Workman pattern. For the fermentation, let's say 3-lactate will oxidize, become pyrite. There will be 3-lactate will be released, and there will be three products. Three pyrite. The pyrite, one of the pyrite will oxidize, become acetate, and then the other two will be stoichiometrically. Other two will be undergo reductive pathway, forming two propionate. So when we balance it out, we will be getting 3-lactate with forming two propionate with acetate, forming carbon dioxide, water, ATP, and hydrogen ion. As a conclusion, let me show you guys this thing. The fermentation of aspartate during propionate acid fermentation is 2-lactate with 2-aspartate with ADP with phosphate forming acetate 2-succinate, 2-NH3 with ATP. So moving on, this is the overall overview of the citric cycle, TCA cycle in other words. So carbon dioxide, this is how it formed, and then let's move on to the conclusion. So as a conclusion, PAB benefit analytically from aerobic respiration with oxygen as a terminal acceptor. There is a low level of oxygen supply which deploys the fermentative behavior on lactate via Wood-Workman cycle. It is to oxidize this fermentation product, products propionate and acetate. In the same time, it pointing out to an extension of its metabolic repertoire, the whole body of items which are regularly performed. There is a production to consumption of propionate, which is also known as propionate switch, to acetate switch, as it described in E. coli. That's all.
Hi, Doctor and my fellow friends. I'm Kasturi here. Let me summarize briefly what we have looked at. So in this study, we have shown increased biomass formation of 4 species of food-grade propionic acid bacteria when they exposed to oxygen and implicating functional respiratory system. So when using an optimal microaerobic condition, Propionibacterium prudentia consume lactate to produce propionate and acetate initially. When lactate was depleted, propionate was oxidized to acetate. So we propose to name the switch from propionate production to consumption in a microaerobic condition as propionate switch. When propionate was depleted, it is known as acetate switch. So it results in complete consumption of acetate and produce carbon dioxide. Both growth rate on lactate and biomass yield increase compared to anaerobic condition. So for the proteomic analysis, it reveals that the abundance of oxygen involved in aerobic and anaerobic electron transport chain are not significantly different between anaerobic and microaerobic conditions. So this implicates that Propionibacterium prudentia is prepared for utilizing oxygen when it comes available in anaerobic condition. So that brings me to the end of our presentation. Thank you, Doctor and my fellow friends, for your attention.